while we're kind of plugging in, I'm going to be talking about biosimilars. So I'll tell you, it's a little bit of kind of your lovely lab-based work. I'm talking about moving on and the clinical development of biosimilar products, of which many, the interest is now being generated are antibody molecules that are coming off patent. So if you've never heard about biosimilars before, I'm going to give you an introduction, then ramp it up, and so you can see some of the challenges that come from my perspective, which is the clinical development of such molecules. No. We'll get there eventually. That's me. Okay. Right, so challenges in demonstrating biosimilarity and interchangeability of biosimilar products. So, as I say, I'm going to talk about biosimilars and why there is so much interest in biosimilars. Then I'm going to talk about the principles of demonstrating similarity between molecules, quality attributes, non-clinical animal, PK/PD bioequivalence, and phase 3 equivalence, looking at efficacy, safety, and comparability. It will hopefully make sense to you as I progress through the talk. And then if there's sufficient time, I was going to talk very briefly about interchangeability, which is a kind of a hot topic within the biosimilar development world. So first of all, what are biosimilars? Well, they are biopharmaceuticals, so they're made in living organisms, usually either prokaryotic, eukaryotic, but there are some exceptions where you can actually use plants. I know of one rice-based system and another one in sugar beet. So it's not always bacteria or eukaryotic cells. And biosimilar medicines are basically versions of existing and approved biological products. And I say versions because we're not saying they're identical. They are similar, hence biosimilars. Because to be honest, you compare to a generic molecule where it's a very simplistic molecule, you're never going to make something like an antibody. You're not going to make it identical due to the folding, etc. So there is that acceptance that you're talking similar. And then you come into argument of, well, how similar is similar? And that is what all the regulation and the guidelines are about. So the reason for the development, of course, it has to be market-driven. And this is the 12 top compounds in the kind of health expenditure. And you can see that a lot of these are indeed antibody products. So if we can make biosimilar products that are basically cheaper, just the same, well, I shouldn't say same, similar, and that will then save the healthcare system significant amounts of money, and it does work. So now these products are coming off patent, and hence now we see the drive to develop biosimilar products. So as they come off patent and are nearing patent, then we see the research and development companies gearing up through their manufacturing, their phase 1 and phase 3, and then marketing approval, ready for launch when the patent expires. So, as I say, potential value of biosimilars is that because of the increased drug, am I pressing the wrong button? Am I holding it the wrong way? Ah, there we go. So biologic therapies are very expensive. They are effective, but they cost. And because of that, there is limited patient access to these drugs. So cost reductions are a significant driver to increase use of targeted biological cancers. Significantly more patients will be treated as biologics fall, and there has been estimations for EU savings. And this is all very real. This is coming into practice. Obviously, there is a lag between the development and the actual drugs coming into market, and then actually seeing that cost-saving effect. So, as I described earlier, the generic approach is not really appropriate because of the simplistic generic molecules like aspirin. You can go to Boots or whichever other shops are available, but you can go to anywhere and you can buy different brands of paracetamol, the generics. They're simple molecules, but that does not work in the biosimilar world. The biosimilars world consists of a stepwise comparability approach. And I'll explain the kind of the pyramid, which some of you may have seen, the FDA pyramid, which is a lovely kind of representation of what needs to be done. But global similarity is all based on comparability between your originator product and your product. So it's a bit like yourself with your antibodies. Which one was the first one? And, you know, doing your similarity and check. So that's why I was smiling to myself, because it's like an earlier and more simplistic, and you're suggesting naming of molecules, etc. It all kind of comes around. So we have global similarity, and it's all based on quality, which is your manufacturing and chemical analysis, your efficacy and safety. Everything, though there are kind of overarching rules and there are product-specific guidelines, everything is still done on a case-by-case approach. No two molecules are the same. So just because you have one company making a biosimilar product, the next biosimilar product is not going to be identical to that similar product. They're all similar. So everything has to be done on a case-by-case basis. And pharmacovigilance at the end of the day is critical. So when you come to your end and you'll come to your marketing, pharmacovigilance and reporting back, it is essential to the development of a biosimilar product. So demonstrating similarity. This is the basic principle as defined by the EU guidelines. There are differences in these guidelines, but guiding principle of biosimilarity is to establish similarity between the biosimilar and the reference product by the best possible means, ensuring that previous proven safety and efficacy of the reference medicinal product also applies to the biosimilar. We are not characterizing new molecules. These are biosimilars. So the focus of any research and examination is on similarity, not characterization of the molecule per se. Now this is where we have the problem. How similar is similar? These are batches of one reference product. And you can see that in natural manufacturing, between different batches of drug, you will get variation. There is no such thing. When you buy your drugs, they are not absolutely reproducible. There is always an upper and lower limit margin of acceptance. So this is the problem. You're trying to compare your biosimilar product to something that keeps moving. So there is no defined kind of absolute value that you have to match. So the first thing you have to do is demonstrate similarity by quality attributes. When I say the quality attributes, these are your kind of markers. So you're looking at what is the attribute, what is the particular characteristic of your molecule. You can't absolutely examine every single nature of the molecule, so you choose particular key attributes of that molecule and compare those, and that's where your biosimilarity starts. And this is your pyramid, your comparability pyramid. And you can see the foundation of it, where the most work is done, is at the structural and functional characterization of your molecule. Then you come up to the lesser human PK/PD, clinical knowledge, animal, and actually the last phase 3, clinical in man, is actually the lesser information, because you're looking at mainly down here, the structural and functional characterization. So as I said, the quality attributes, you're trying to figure out how similar is my molecule at the basis of your starting points. You've got your cone, you're expressing your products, you're starting your manufacturing, And as you might well know, as you scale up through manufacture, nothing's ever quite the same. What you do in 2 litres is not necessarily the same as what happens at 1,000 litres. So you're always having to go back and check your quality attributes. You're having to set your goalposts. So you yourself are setting those margins of what is acceptable. Is it above, below those margins? And you don't just take one batch of the reference product material. You might take, say, 5 batches. Because if you choose a batch that's down here, and you're comparing your molecule down here, well, actually, in the 5, that happens to be the lowest one. All the rest are up here. You're then setting your margins wrong. So you have to do quite a lot of background work on setting those margins, because they are going to affect all your downstream kind of reference and your goalposts as to what's acceptable. And the FDA now do what they call a fingerprint-like analysis algorithm. So they are looking at, for you to specifically look at the key attributes. They're not expecting everything. They want the fingerprint. What are the important parameters that you're looking at? And from all of this then allows you to then look at what is different about my molecule. Not really what is similar, but actually what's different. Because now I'm going to have to go and look at that difference in the animal models and in the human, and make sure we explore that. You can't just sweep it under the carpet and just say, oh, well, that was a bit different, but everything else was the same. You can't do that, because that one little difference might then go on to have a dramatic effect in your patient population. So with non-clinical animal comparability, again, it's on a case-by-case, dependent on your outcome of your quality data. The EU applies the principle of the 3 Rs. We like to look after our animals. We don't really like to kill our bunnies if we don't have to. So in the US, they are more strict. They do like to do still the non-clinical work. There is very much questions surrounding non-clinical, for example, immunogenicity. How relevant are the animal models with regards to rates of immunogenicity later in man? So it is a balance as to what is required, but it's a good indicator before moving into man. So bioequivalence. Bioequivalence itself is based on really the generic profile. We set the confidence intervals between looking at your reference product and your product. They're always set between 80 and 125%. Nobody actually quite knows where this 80 to 125% confidence interval and margin derived from, but it's what's been inherited from the generic world. So you have to make your product, when you're testing it in your clinical trial, looking at bioequivalence, fall between that 80% and 125. So it can be less and it can be more, but those are your margins. So as I say, with the generic world, that might be quite easy to hit that upper and lower margin. But in the biosimilar world, with biologic products, it's more problematic. And because of that high variability due to the complexity of the molecules, you then require larger phase 1 studies, about 150 to 300 healthy volunteers. Generic products, you're looking maybe 30 patients. So you can see the increase in the amount of work that has to go into it, the cost, etc. And it can be actually, for healthy volunteers, that's good. Some products actually require patients. So trying to find patients that fulfill your criterion can be a challenge in itself.
Would that still be considered a phase 1 participant?
Yep.
Okay.
That's phase 1, yeah. So then moving on to the phase 3, I don't know if people know the kind of phase 1, phase 2, phase 3 development. Phase 2 is usually when you look at your dose, but we don't do that in the biosimilar world because we are using the dose based on the originator product. So we can skip over that one. It doesn't mean that we're doing any less work because we've done a lot more characterization than probably the originator product. And we do a lot more post-marketing work of following up and checking. but it just means we can hop over that because we are matching the dosing regime of the originator product. So when we come to look at biosimilarity in the phase 3 studies, which is the larger studies, we'd have to look at the patient population and what we call assay sensitivity. Now, what do I mean by a sensitive homogeneous population? Well, we have to have a clinical trial in a population which is going to show the right results. Now, just because I fancy from a marketing perspective doing a study in rheumatoid arthritis because I know that will sell my drug. I know that I can recruit rheumatoid arthritis patients quickly and easily, and I have lots of clinics I can go to. Not necessarily, because that might not be the most sensitive patient population. So the authorities might well deem that I have to go and do it in some psoriasis patient population, which is harder to recruit, which will take longer for me to find, but is a more sensitive patient population. So you can't just take necessarily the easiest and the most kind of commercial route in development. So the endpoints, likewise, aren't necessarily the endpoints that the clinicians are used to, because they have to be the endpoints to show sensitivity and show difference. Even though we're looking at similarity, it has to be the endpoints that will show and highlight the difference. So margin effect size, as I say, and appropriate margins have to be set. And the smaller the margin, the narrower the confidence, basically the larger the study, which is similar to what I described earlier. There is a ICH E10 guideline, but I'll summarize it. Basically, it says that you have to use the data you're setting your margins from is based on historical data from the originator product. To base that number, say, okay, I know I'm going to get an effect size this size, and my product is going to fit within that effect size. You have to match that original study. It's like you doing variations in your test tube, changing all the reagents and expecting that you're going to get the same result. You have to base it on what the assay was used before. So this can cause problems when designing studies, because what happens if we call the standard of care has changed? You're now using better chemotherapy regimes than what was used originally on the basis of your product. You can't go to clinics and hospitals and say, okay, we want you, because this fits our clinical trial, to use the old chemotherapy regime that isn't as effective, but then it's nice because we can then prove our study, that's improved 15 years down the line. So you really do have these problems where you're trying to kind of work around historical data and moving things forward. Now, you can get around it by statistical wizardry, which I'm not good at, but what you can do is say, actually, well, the new chemo regime is 10% better than the old one. I know the effect size of the old one was 10%. Therefore, kind of the overall difference is actually going to be 20%. So you can sometimes do that kind of like extrapolation downstream, but it is very, very difficult and is not always clear. And sometimes you have to go to the authorities, and we have to take scientific advice and negotiate these steps. Equivalence or non-inferiority. I was describing to you that we have the upper and lower margins. Now, when you're doing a clinical phase 3 study, you are, in some cases, you normally have to look at both ends. Is my drug less effective, or is it even more effective? Because more effective isn't necessarily good, because it can affect your dosing of the patient. So you actually have to stay within the margins. So more is not necessarily good, less is definitely not good. So what you can sometimes do is, depending on the drug and knowing that, okay, if it is more effective, that's not going to affect my patients, you can then say, okay, I'm just going to do the focus on what we call a non-inferior study, that my drug is no less effective. That then means you can actually make it a much more simple study, and your size of the number of patients you're recruiting comes down. So it's another parameter that needs to be looked at. How am I doing for time? Can I do interchangeability?
Yes.
Right. Interchangeability. As I say, moving on from clinical development, it is a hot topic in the biosimilar world because it all comes down to, I'm afraid, the marketing aspect. Biosimilar products. If you have a new patient coming into your clinic and you have approved biosimilar and you're happy to use that biosimilar as a clinician, you can say, right, you can go on to this drug, it's a similar product. But interchangeability is where you've got patients like, for example, patients who have rheumatoid arthritis and are on long-term medication. Can you have a patient that's been coming in for 6 months, 8 months or whatever, stable on their medication and switch them? So this is called the switching. So interchanging the products. And this is where the market is based. There really is. This is the kind of the golden ticket for biosimilar products because according to the EU, once you have biosimilar approval, it's interchangeable. They should be identical. Not that easy. So in the US, according to the US Act that allowed for biosimilar approval, which has only just happened in the past few years, the EU were well ahead of the US on this. I'm very proud of the EU for that. It just should be a drug company, but in the US legislation, they rather made things very complicated for themselves because they put in this little clause that say the drug company has to show that nothing happens to the patient if you switch them back and forth. Not just patient population, you're actually talking on the individual patient basis, which, you know, how are you going to prove it down to that level? But that is what their guideline says. They also have absolutely provided no information on how you're meant to do this. And everybody is kicking their heels, waiting for them to actually provide that information. But the FDA haven't quite got around to it yet. I think they're trying to figure out the best way around. So they've rather tied themselves up in knots on that one. But meantime, even if the FDA approve a drug and say, yes, we approve it for interchangeability, all the states in the US are now running around and the originators are doing whatever they do, kind of going to the various states and saying, we don't think this is a good idea, and they're all having a bit of a hoo-ha, and the local states, the states are passing rules saying, no, we don't want substitution. We're not going to allow it. So you might have a drug that's approved by the FDA. Yes, you can do it. But at a state level, they're saying, no, we're not going to allow you to do it, which is a rather conflict of kind of the FDA, the government body, saying one thing and the individual state saying another. And this is literally, this map changes from month to month. I can never keep up, but that was the latest one about what the individual states are going around. In the EU, it's kind of a similar situation, maybe not quite as in-depth. The EU, the EMA says, yes, you can, but it's down to the individual countries to make their own rules, basically. So you do have some, but a lot of countries are saying, no, we don't want interchangeability. We don't want automatic substitution that the pharmacist can say, okay, I'm substituting this one for a cheaper version. So again, that's kind of a similar map within kind of Europe as to the kind of the complexities of the different regulations that allow it. Australia and the Finnish agencies have actually kind of done an about turn, and they're actually now coming more out on the side of saying, yes, actually, do you know what? We recognise biosimilars absolutely as substitute, and they can be substituted. They are similar products, and they are safe to do this. So this is where we think the barrier is beginning to break down, but there's still a long way to go. Just a quick slide to say that to do the interchangeability, you're going to have to look at all these endpoints, duration of evaluation, response and margins, things that I've described earlier. So with regards to kind of interchangeability, the big elephant in the room is interchangeability, and whether we're going to consider a biological product more like a new molecular entity, or a new product with similar function, or are we going to see it as biosimilars are interchangeable? Okay, that's it. Well, I hope you've learned something about biosimilars.
Well, thank you.
If anybody wants the guidelines, they're the guidelines.
I must have missed something because of your description of a certain level of variability from batch to batch within the margins that you require implies that you're not dealing with one single molecule, you're dealing with a mixture of molecules, and that confuses me.
I can't go, I mean, I'm not an expert about what the content of, say, rituximab, etc., whether it's polyclonal or kind of monoclonal,
no,
etc.
Talking about antibodies, are we?
Yes.
Only antibodies?
No, not only, but for the most part.
But any manufacturer, you will get variability between batches. You will. It's just, that is life. And then if you do a change in manufacturing, oh my gosh, you get a whole kind of shift. And if with that, you then actually have to go back to the drawing board and prove equivalence again to your original way you were manufacturing your product and do comparability. There is, believe me, those margins exist, whether it's GCSF, EPO, protein, etc. You will get that batch-to-batch variability. If you look at any of the data sheets, you always have a higher and lower margin. They're never the exact value every single time.
I recognize there's batch-to-batch variations purely down to manufacturing, but that would be reflected by the concentration of the product if you're dealing with a single molecule. If you're dealing with a mixture of molecules, then that variability goes up by orders of magnitude. Because then you have a mixture and you don't have under control which molecules within that mixture will have the highest concentration and which one has the lowest concentration. And that will vary from batch to batch.
Yeah, and I can imagine that being the case. Because, for example, with low molecular weight heparins, it is a mixture of types of molecules.
Exactly.
And that is what, when you go to the doctors and get treated, you will have that variability within your processes.
So what I was not clear on, and that is now becoming clear, is that biosimilars mostly means a mixture of molecules. It's not a single defined molecule that is going to be ideally interchangeable with the original drug, which was a single molecule.
No, I can't tell you enough about they are all based on a single clone.
Not all the biologics are a single molecule.
Yeah.
We do have chimeric products, and that's another issue that I was going to ask you about. The problem with the biologics that we are currently using, and intensively using in rheumatology practice, I'm a rheumatologist by the way, that after a time we face development of antibodies to these drugs. So what about biosimilars? You're speaking about a combination of molecules together. You have a well-defined molecule that you're building another similar molecule to it. So
Yes.
It's a bit of a threat rather than a treatment to us, because I always face problems with my patients. I have to switch. If you revise the ACR guidelines and the European guidelines, you'll find that all the time there are updates for the guidelines to give you space to move between the biologics within the same disease category because of the threat of the antibodies, whether human or whatever. So with the rituximab, for example, it's a chimeric, it's a human murine molecule. So, and now we're having a biosimilar in pipeline for rituximab, I think, by Biogen.
Yeah, no, very much so.
Yeah.
And I mean, you have one for the infliximab. So that's where the pharmacovigilance comes in the post-marketing safety review. And I mean, there is going to be, specifically with the antibodies, that is going to move more into the post-marketing side of things, where because some of the effects, you will be seeing the immunogenicity, etc., and especially if it affects efficacy, because you can have an immunological response that has no effect on the patient, but you exactly, and not be a neutralizing antibody, is those percentages are so small that you can't detect that in your clinical trial, because you're looking at a patient population of maybe, well, they can vary from 300 to 1,000 patients, but if you're looking for percentage effect, it might be around about 4%. So, and of that, You might have no cases detected of neutralizing antibody, you're just detecting antibody. So you're going to have to move that into the post-marketing kind of reporting system event. And that's where pharmacovigilance reporting is very important in this side. And nobody says that that element is not required.
Actually, we're seeing a lot about biosimilars, and I think we need more from you regarding this, because our background as rheumatologists regarding biosimilars comes only from papers that are published, and we are not very well convinced about it. And actually, we feel rather afraid to take the step of using the biosimilar in our patients in practice. So I think we need to know more about biosimilars.
I mean, in a way, I'm surprised that you haven't been visited by the sponsor companies, because usually they provide you with kind of the background information regarding their kind of products, especially if one is being launched. So I'm sure you will be visited when Rituximab is launched. You will be obtaining a lot. But if you need any information, the EGB is a good place to go for information regarding kind of biosimilars. And I'm very sorry for anybody who's purely lab-based. I hope that was of some kind of interest and introduction to you.
Well, thank you, Hazel. I think it certainly was. You know, I'd like to make a brief statement regarding the molecular end of your question. First of all, within the body, the distribution of an antibody, a valuable antibody, there's always a distribution of microheterogeneity. So the parent focus, the parent entity that the originator looked at was a distribution. Their products are definitely distributions. There's microheterogeneity. There isn't one species of molecule being produced. So now we're looking at biosimilars that are trying to match a distribution. And the question is how close does that distribution have to match? And in terms of what types of misrelation, glycosylation, what elements of that microheterogeneity have to be addressed? And it's a big topic. And
that's why I was saying the focus actually is not on how similar my molecule is, it's how different my molecule is. And that's what you're looking at, is those attributes and the focus and tailoring it.
Differ from the original drug then. If the original drug was already an antibody, then the biosimilar, and it would have the same characteristics, the same variability as the biosimilar does. So it wouldn't make any difference in terms of quality control between the original drug and the biosimilar.
But you have to say the original distribution, because there wasn't one entity, and that's now. And one more factor in there is to talk about process-related impurities and process-related entities. Because not only in the parent model, but in manufacturing then, breakdown products or non-standard entity molecules can be designated as part of the entity or not, depending on definition. So it's a big topic, but thank you so much. We've got a great introduction, and I think I'm going to be told to get going. Okay, so we're going to break for lunch on time, and then just.